Hello, everyone. Thank you all for joining us for our live webinar, which will be presented live by our guest speaker, Sean Davies, and entitled The Role of N-acyl-ethanolamine Biosynthetic Enzymes in Attenuation of Cardiometabolic Diseases. My name is Maria Fedorova, and I'm based at the Technical University of Dresden. The webinar will be around 45 minutes, allowing 10 minutes at the end for questions. So let me introduce our speaker. Dr. Sean Davies is Associate Professor at the Department of Pharmacology and the Division of Clinical Pharmacology at Vanderbilt University. He did his PhD study at the University of Utah, examining cell signaling pathway induced by oxidized phospholipids. He then did postdoctoral studies with Dr. Jackson Roberts at the Vanderbilt University, studying the role of reactive bicarbonyls generated by lipid peroxidation, especially isolevoglandins, in chronic diseases. During this time, he identified 2-aminomethyl-phenols as effective small molecule scavenger for decarbonyls, and subsequent studies have demonstrated the efficacy of these scavengers in animal models of chronic diseases, including atherosclerosis. In 2007, Sean received one of the inaugural NIH Director's New Innovation Awards, and this funding allowed him to demonstrate that incorporating probiotic bacterial strains specifically bioengineered to generate the high levels of N-acyl-phosphatidylethanolamines as a precursor of N-acyl-ethanolamines into the host gut microbiota could be used to treat cardiometabolic diseases. More recently, studies in his lab have focused on understanding how the biosynthesis of N-acyl-amine is regulated in vertebrates, their role in regulating metabolism and inflammation, and the potential of small molecules activators of NAPE-PLD to ameliorate cardiometabolic disease. So I guess we will hear more about that. And welcome, Sean, please.
All right. Thank you, Maria, for that introduction, and I really appreciate Lauren and the organizers of this Lipid Maps webinar for giving me the opportunity to speak. So my lab is interested in understanding the fundamental drivers of three closely related cardiometabolic diseases, and these include atherosclerotic cardiovascular disease, which is the major, the leading cause of death in the world and certainly in the United States, type 2 diabetes, which affects about 11% of the US population, and metabolism-associated steatotic liver disease, which used to be called NAPLD, which affects about a third of the US population. And the reason we want to study these together is they have overlapping risk factors, including things like obesity and oxidative stress, dyslipidemia, and inflammation. And so our thinking is if we understand the mechanisms that drive these combined risk factors and can find ways of treating and preventing that, we can actually treat or prevent all three of them. And my lab focuses really on two different types of bioactive lipids. Half of the lab looks at bioactive lipids that are generated by lipid peroxidation, and these are the reactive carbonyls, and I'm not going to talk about them very much at all today. But the other half of the lab has started focusing on the set of protective lipids, the N-acyl-ethanolamines, which I'll refer to as NAEs. And I just want to give you the basic framework that of how we think about these, and then I'll go through various data from other labs that sort of helped establish this framework, and then show you data from our lab that helps, I think, flesh out some of this. And one of the things I want to emphasize is how much is unknown within this framework. The basic idea is that in response to cellular stresses and cellular injury, you have the biosynthesis of these NAEs, and then they induce responses that are protective to help drive the cells and the tissues back into homeostasis. And as long as this is functional, it helps to prevent the onset of cardiometabolic diseases. But then under obesogenic and atherogenic conditions, you have a reduction in the ability to do biosynthesis, and so you lose these protective responses. And then another part of our hypothesis is that pharmacologic administration of PEA and OEA can restore protective responses, or what we're doing more recently is trying to target the enzymes that are actually making these and restore that so that we can restore these protective responses. So that's the overall framework that I'll be talking about today. So there are many different N-acyl-ethanolamines, but I'm going to focus on the three that have been most studied. These are palmityolethanolamide, PEA, oleoylethanolamide, OEA, and arachidonyletanolamide, which I'll refer to as anandamide. So these different NAEs act on various receptors, and this can account for much of their bioactivity, but they have different affinities for these receptors. So for instance, anandamide is a potent partial agonist of the endocannabinoid receptors like CB1, but OEA and PEA are not. PEA is a potent agonist for GPR55, which is sometimes referred to as the third endocannabinoid receptor. Anandamide also has reasonable potency, and OEA has at least some potency. OEA is one of the most potent known endogenous ligands for the nuclear hormone receptor PPAR alpha, and PEA and anandamide can also act as ligands. OEA is also a reasonable agonist for GPR119. So again, based on these receptors, this can account for much of the bioactivity that we see, but they also have other targets that they can act on. So the biosynthetic pathway for NAEs is somewhat more convoluted than you might imagine, and why this is so, why evolution chose this pathway for vertebrates is unclear. It's not simply the condensation of an acyl-CoA with ethanolamine. Instead, the acyl chain typically is coming from the SN1 chain of phosphatidylcholine, and then it's added to the nitrogen of phosphatidylethanolamine through enzymes called PE N-acyltransferases to make this unusual phospholipid, the N-acyl-PEs, which I'll refer to as NAPEs for most of the talk. And then you can have hydrolysis of the NAPE to form the N-acyl-ethanolamines. And it's even more convoluted because, for instance, in humans, there are 6 different PE N-acyltransferases, 5 that are calcium independent, Plat 1 through 5, and 1 that is calcium dependent, PLA2 group 4 epsilon. Then for the conversion of the N-acyl-PE to Ne, there are multiple pathways. The most direct pathway is the NAPE-phospholipase D, so I'll refer to as NAPE-PLD. And this directly hydrolyzes to generate the NAE, but also phosphatidic acid. But there are at least 2 other additional pathways where you use multiple enzymes to generate the same compounds. And then these can be catabolized by Pha and Na back to the original fatty acid and ethylamine. And we really do not understand the complexity of the system, which one of these enzymes are involved for many of the biosynthesis that we see. And so this is what my lab is really interested in trying to understand. So I just want to give you a brief history of several of these biosynthetic enzymes. And one thing to keep in mind is that many of these were only fairly recently characterized. The one that was characterized the longest ago was NAPE-PLD. This was work done in 2004 by Natsuo-Ueda's group. And this gene is highly conserved from worms to humans. It's expressed in a wide number of cell types and tissues, so it's a fairly ubiquitous enzyme. Structurally, it's a metallo-beta-lactamase fold. It acts as a homodimer. It's a peripheral membrane protein, and it actually associates with many, many different membranes. So the Golgi apparatus, vesicles, plasma membrane, nuclear membrane, early endosomes. And again, we don't really understand what this localization means biologically. It's not currently known to be part of a protein complex. It doesn't have known binding partners. It's constitutively active, although it can be hyperactivated. And it's important to note that this is a very different phospholipase D from the classical PLD1, PLD2. It doesn't do transphosphatidylation. It's a zinc hydrolase. And it does not act on PE or PC. It acts on these N-acyl-Ps, and it needs to be at least N-acyl-Ps of at least 4 carbons or longer. But once you get longer than 4 carbons, it really doesn't seem to have a lot of selectivity. In terms of the calcium-independent N-acyltransferases, these have been renamed to phospholipase A acyltransferases, so PLAT, and there's PLAT 1 through 5, with PLAT 2 having the most acyltransferase activity, PLAT 1 following that, for a little bit of activity, in 5 and 3, very little. 1 through 4 are integral membrane proteins having the structure that's shown here. The most characterized is really PLAT 2, We don't really understand how its activity is regulated. It's not calcium dependent, but it's also not dependent apparently on pKa or pKc. And there's a wide distribution of these plats through many tissues. And again, we don't really understand how this controls which NAEs are formed and why they're formed and such. The calcium-dependent N-acyltransferase was the last to be characterized, even though the activity was known for like 50 years. So Ben Cravat's lab took brain lysates, fractionated it, and were able to show that there was a correlation between the acyltransferase activity and PLA2G4 epsilon. Then when they took the recombinant enzyme, they were able to demonstrate that it was indeed calcium dependent and that its acyltransferase activity far outweighed its PLA1 and 2 activity. So even though they used brain to isolate it, if you look in the Protein Atlas database at the mRNA expression, really the one tissue that really lights up in terms of expression is skin, and there's smaller amounts in the oral mucosa, tonsils, kidney, and cervix, and then relatively very, very low amounts in the brain and lung, and yet we know it's important in the brain. So again, we don't really understand much about how expression correlates with that. Okay, so now I want to just talk about some of the data that support this basic framework and then go into the work that my lab has done. So what's the evidence supporting that cell stress or injury induces the NAE biosynthesis? So way back in the 1970s, Harold Smith's lab showed that in myocardial infarction, you had this massive 50-fold increase in the NAEs in the infarcted tissue. And then later on, they showed the same was true in the brain with ischemia. They got these massive increases in both NAPEs and NAEs. Then looking at less catastrophic stressors, just like, for instance, UV irradiation, here you see about a 4-fold increase. And then a number of other of these, what I'd call sort of homeostatic stressors, also give these small changes in NAPE and NAEs. So for instance, feeding, which of course increases the level of free fatty acids fairly dramatically, will increase OA levels about 3-fold, canic acid, norepinephrine, PKC, PKA activation, histamine, all of these will give about 2 to 4-fold change in various NAEs. So there is at least this patchwork of knowledge that some stressors are inducing the NAEs, but we really don't understand the mechanisms. And I think the one exception to that is now with catastrophic stress. Ueda's lab showed in 2022 that the PLA2 group 4 epsilon is likely to be the N-acyltransferase that is responsible for formation of NAPEs in the ischemic brain. But for the mild stressors, we really don't understand the mechanisms, and we don't know which enzymes are actually involved. And we really don't have an overall framework to understand the universal, is this response of NAE biosynthesis a universal response to stress or not? So it's a data that suggests that if you have these PA and OEA, that it's actually inducing protective responses. So I'm going to start with PA because it was the first of the NAEs that was actually characterized. This started back in the 1950s, where people were looking for an anti-inflammatory factor in eggs because it was known that children that had an egg-poor diet seemed to be more susceptible to rheumatic fever. And so in 1957, PEA was identified as the anti-inflammatory factor in egg yolks. Later on, it was shown that PEA would block mast cell degranulation, and this is where the term autocoid local injury antagonist amides was coined. Now that's a mouthful, but I think it really highlights the key idea that these are generated in response to injury, and then they help to limit the damage from the injury and to return to homeostasis. In 1998, PEA, but not OEA, was demonstrated to inhibit pain behaviors that were induced by formalin. And in 2008, PEA and OEA, but not an anandamide, was shown to inhibit carrageenan-induced paw edema. So all of these suggest an anti-inflammatory effect of PEA. Later on, I'll also talk about a pro-resolving effect. I'm not going to talk about anandamide. Obviously, Raphael Mechoulam's group identified this as the first endogenous ligand for the cannabinoid receptors back in the 90s. the key point I just want to make is that in general, the peripheral levels of anandamide are far lower than PEA and OEA. And also of interest to us, many of the stimuli that alter the levels of PEA and OEA, they either have no effect on anandamide or they actually drive anandamide in the opposite direction. And again, because these NAEs share, at least theoretically, the same biosynthetic pathways, it's unclear how this happened. So the last of the NAEs that were sort of well characterized was OEA, and this is work by Daniel Piamelli that really showed that it was an anorexic lipid. So when they were doing IP injections of various NAEs, and I suppose they were probably looking mostly for the effect of anandamide, and not surprisingly for something that activates the cannabinoid receptor, it increased food intake. But OEA markedly decreased acute food intake, and rather potently, PEA also did as well, but less potently. Oleic acid didn't have any effect. And I'm just showing some of the data here, but they showed that this IP injection of OEA lit up the areas of the hypothalamus responsible for controlling feeding behavior. And they also showed this wasn't just a pharmacological effect. If they were measuring OEA levels in the small intestine, in a fasted animal, those OEA concentrations went down, and then they increased in response to refeeding. So this was actually an endogenous response. Subsequently, other labs like Harold Hansen's lab showed that you had the same rapid increase in the N-acyl-PE levels, and these basically predicted which NAEs would come up. Piamelli's lab showed that the induction of OEA was extremely rapid, so even within 10 minutes of refeeding, you had this bounce in OEA levels, which would strongly suggest that this isn't an increase in enzyme expression, but rather a rapid increase in the biosynthetic enzyme activity. With Gary Schwartz and Piamelli's lab together showed that the driving factor for activation of PEA biosynthesis was fat, and that, for instance, if you gave oleic acid but not palmitic acid, that you would get the increase in OEA, and that you got the increase in N-oleo-PE. They also showed that oleic acid increased NAPE-PLD activity and decreased the activity of the hydrolyzing enzyme Pha. So perhaps as importantly, Piamelli's group showed that OEA was a direct PPAR alpha agonist, so it bound to PPAR alpha, it drove gene expression. The effect on food intake of OEA required PPAR alpha because when it was deleted, you lost that effect. And not surprisingly, for a PPAR alpha agonist, it increased fat oxidation. And this was dependent on PPAR alpha. And I think as importantly, the effect of OEA on food intake was not the driver of changes and reductions in triglyceride, because if they did pair feeding to match the reduction in food intake, this did not replicate the full effect of OEA on reducing triglyceride levels in various tissues. And I point this out because, again, as we think about inflammation, there's the general notion that glycolysis sort of drives pro-inflammatory phenotypes and macrophages, and that switch to oxidative phosphorylation tends to drive resolving phenotypes. And so activation of PPAR alpha is likely to be pushing us toward a resolving effect. Pharmacologic administration of OEA worked in obese animals, and as importantly, could also reduce cholesterol and triglyceride levels in the serum. So this strongly suggests that OEA and PA do invoke protective responses that potentially protect against cardiometabolic disease. And this is really where my lab started to be very interested in this. And the reason why is because I had received this new innovator award from the NIH director. This was the first cohort of these high-risk, high-reward grants. And what we had proposed was basically that we could use engineering of gut bacteria to produce therapeutic compounds that we could then colonize the gut with these bacteria, and then this would serve as a way of chronically providing therapeutics to treat chronic disease. And our original plan and what we had proposed was primarily peptides like ApoA1 mimetics and GLP-1, but we weren't having much success with that. And so we started to think, well, it might be easier if we were having the bacteria generate small molecules, metabolites, things that were protective. And during this time, Jerry Schulman's paper came out that showed that IP injection of the N-acyl-PE was as effective as what we've seen with OEA, and interestingly, the N-palmityl-PE would have this effect, and that it was still effective in obese animals. And so we knew that bacteria could actually produce small amounts of N-acyl-PE on their own, and so we thought, well, let's just juice up their ability to produce that, and then they'll be secreting it, and let's see what the effects would be. And so to do this, we used a probiotic bacteria strain, N-acyl 1917, and transformed it with NAPE synthase from Arabidopsis. Now, the reason we chose this is that at that time, none of the mammalian P N-acyl transferases were cloned, so this was the only gene that we knew of that we could transform to increase NAPE synthesis. The other thing I want to point out is that E. coli, their primary phospholipids are PE. So essentially, when we're giving N-acyl that have the empty vector transformed into them, we're essentially delivering PE to the animal. And so if we took these bacteria and we either gave them by gavage, or we gave them in drinking water, and we looked at the levels of the NAPEs in the colon, where they colonize, we could get increases in NAPE levels. And so this got us excited and allowed us to ask the question, if we have some way of chronically administering these NAPEs, will we get protection against obesity on a high-fat diet? And so the way we did these studies, first to enhance colonization, we gave ampicillin to the animals sort of to clear out some niches that would allow colonization. And then we gave the bacteria in drinking water for 8 weeks. And so our base control was just a high-fat diet with standard drinking water. Another control was the vehicle. So this just had a small amount of gelatin that we knew we need to put in the drinking water to keep the bacteria happy. We also did the empty vector control N-acyl in the drinking water. And finally, we did the NAPE-expressing bacteria. And so we gave the bacteria for 8 weeks. And then because we were looking for colonization, we stopped administering the bacteria and just followed them on the high-fat diet for another 12 weeks. And I'm just going to focus on this part. Not surprisingly, if you put a mouse on a high-fat diet, they rapidly gain weight and they rapidly gain fat. And if we gave the vehicle or the bacteria with just the empty vector, that basically didn't change the trajectory at all. So just having bacteria in drinking water doesn't affect the mouse's behavior or such at all. But if those bacteria are now expressing NAPE, we see that there was this significant decrease in that rate of weight gain and particularly the rate of fat gain. So it was protective. And when we looked in the liver, we see a significant increase in genes related to fat oxidation and a decrease in genes related to inflammation. And we also saw improvements in glucose tolerance. So overall, we are having a protective effect against metabolic disease from this long-term administration, so for 8 weeks. Then, as I said, we followed them out, and when we looked at the bacteria, we could monitor them because they were fluorescent. We saw that the bacteria would persist fairly strongly for 4 weeks, and then they start to peter out. And when we looked at the effects on food intake and fat gain, it persisted for 5 weeks after we'd stopped administering them, and then it petered out. So we didn't get the lifetime colonization that we were hoping for, but we had fairly significant colonization. And as long as the bacteria were producing the NAPEs, we had this strong protective effect against metabolic disease. And I should say, in subsequent experiments, we showed that even if you start on the high-fat diet for 2 weeks before you start giving the bacteria, so that you already have the insulin resistance, you already have the inflammation, you already have that, we still saw protective effects. So all of that was very exciting to us. Now, during the time when we had published this, there was an important question raised, and that is whether it was actually NAPE or the NAE that was the driving protective response against metabolic disease. So when Gary Schulman's lab had done their experiments, they actually thought that, or put forward that it was the NAPE that was actually the most important thing. And again, they could show that IP injection of the NAPE significantly inhibited food intake, and if they infused NAPE into the intestine, they could actually see a small amount of that NAPE make it to the brain. And if they directly injected NAPE into the brain by ICV injection, they could actually dramatically reduce food intake. And if they injected PEA, which was what NAPE-PLD would have converted that N-palmitoyl PE2, they didn't see any protection against food intake. And they also, if they injected PE itself, they didn't see any protection. So they interpreted this to mean that it was actually NAPE that was the anorexigenic factor. But then Harold Hansen's lab, they did what was kind of the obvious experiment, and that's do the same IP injections in NAPE-PLD knockout mice. And they did in fact see that NAPE was still able to provide this reduction in food intake, but they did studies, and this is just part of them, showing that if you did PE injection and also phosphatidic acid of the same concentrations, that also reduced food intake. And so they interpreted this IP NAPE effect as being just an adverse effect of injecting a lot of phospholipid. Now, at the same time, I'll point out that we're somewhat agnostic about whether it was NAPE or NAE, but we definitely would see that when we had our NAPE bacteria, we were getting increases in NAEs in other peripheral tissues. And so we thought we actually have some tools that would help test this. And so I think this is an important way of sort of adding to that overall framework that I'm talking about. And so again, we knew that if we just gave our N-acyl that are mostly producing PE, that there wasn't any adverse effects on the animals from this. And so that helped us to have a baseline control where we weren't getting adverse effects on food intake from it. And then we had our NAPE-expressing bacteria, but then we made a third strain of bacteria, and these were those that were co-transformed with NAPE synthase and NAPE-PLD. And when we did mass spectrometry on these bacteria, you can see that the NAPE-ECN expressed high levels of NAPE, but very little NAE, whereas the co-transformed bacteria express very little levels of NAPE, but high levels of NAE. And so we had this, pretty nice dichotomy between the bacterial strains. And so we did a test in wild-type mice, fairly similar to what we had done before, although we didn't follow them that long after we stopped feeding the bacteria, and looked at the effect on food intake and body weight. And here I'm just showing the fat gain. And what you can see is that both the NAPE-expressing bacteria and the NAE-expressing bacteria were equally good at inhibiting fat gain, suggesting in our minds that the NAPE was being converted to NAEs. But the more important experiment was then we did the same studies in NAPE-PLD knockout mice. And here what we see is, once again, the NAE-expressing bacteria are very effective, but now our NAPE-expressing bacteria no longer have any effect. So we interpret this to mean that at least under these conditions, that NAPE needs to be converted to NAEs by NAPE-PLD to have the protective effects against metabolic. So that's a rather simple model. That's just a high-fat diet, but that isn't really modeling the more aggressive forms of cardiometabolic disease like atherosclerosis and liver disease. And so we repeated these studies now using LDL receptor knockout mice. And these mice will develop atherosclerotic lesions when they're placed on a Western diet. They don't if they're just kept on a low-fat diet. And so we administered the bacteria for 12 weeks and looked for changes in outcome. And in the LDL receptor mice, we were still able to have effects on body fat. Interestingly, we actually didn't see effects on food intake. So again, this sort of suggests that it's the activation of PPAR alpha and fat oxidation that may be a critical response. Another thing that was interesting was when we looked at NAE levels in the peripheral tissues, you can see that simply going on a Western diet markedly reduces the level of NAEs in these tissues, and that if we give the NAPE-expressing bacteria, we're able to rescue these levels of about 50% back to the what would be in the control animals. But did that have an effect on liver disease and on atherosclerosis? So when we look in the liver Not surprisingly, when the animals are on a Western diet, they have significant accumulations of hepatic triglycerides, and this is blocked not by the control bacteria, but by the NAPE-expressing bacteria. There's also inhibition of inflammatory cytokines like TNF and fibrosis. So we did have really very, very nice effects on liver disease. What about on atherosclerosis? So here the effects are quite interesting. So we were able to see reductions in serum cholesterol levels, but this didn't translate to significant differences in the overall lesion size. There's certainly a trend here, but it's not significant. But even despite not actually changing the lesion area, we had dramatic effects on the amount of necrotic core within those lesions. And this is critical because it's this hollowing out of the lesion, the formation of the necrotic core, that's thought to drive the formation of vulnerable plaques. But we didn't understand how this was possible, how we could get this protective effect without changing lesion areas. And as we were trying to get this published and trying to understand what was happening, Sabine Stephan's lab did a very similar experiment and basically demonstrated probably what was going on. So here they didn't use LDL receptor knockout mice, they used ApoE mice, knockout mice, which are also prone to atherosclerosis. And here they weren't using bacteria, but they were just doing daily IP injection of PEA for 4 weeks. And in their experiments, and they did a number of different experiments, so I'm just showing one of the studies here. They first gave the animals a high-fat diet for 12 weeks, so they developed strong atherosclerosis, and then they started the treatments and kept them on the high-fat diet. And in these studies, there's not a change in plaque area, but there's a significant reduction in the necrotic core. And there's an increase in the collagen content, suggesting that that fibrotic cap has been extended and is solid. And the key observation that they made was that this most likely suggested that the PEA was enhancing efferocytosis, enhancing this resolution that was critical to prevent the progression of the disease. So what is efferocytosis? Well, every day we face insults where inflammation occurs and various things. And you have leukocytes that infiltrate into that site. They do their job, they eliminate the threat, and then they undergo apoptosis. But then you've got to resolve that inflammation, and one of the key steps is that resident macrophages or monocyte-derived macrophages, they have to phagocytose these apoptotic cells and get rid of them. And of course, when they take up the apoptotic cell, there's a lot of cholesterol and the individual components of the cells, and for cholesterol, reverse cholesterol transport is a key factor in that. Now, in a healthy person, this happens. Maybe you get a little oxidized LDL and you have a little inflammation, and the leukocytes come in, they become apoptotic, but you clear them. And so you do the efferocytosis, you release the factors that help with resolution of inflammation, and so you just go back to having healthy tissue. But in atherosclerosis, that's not what happens. So there's a lot of accumulation of oxidized LDL, a lot of accumulation of apoptotic cells, and for various reasons, efferocytosis breaks down. And so these apoptotic cells aren't cleared, they undergo secondary necrosis, and they're releasing proteases and other factors, which really hollow out and form this necrotic core. And this can expand over time, and it helps to hollow out that fibrotic cap, and eventually if that thins too much and under stress, it can rupture, and of course you have platelets that aggregate and cause thrombi and cause infarction. So they proposed that the PEA was actually enhancing efferocytosis, and looking for evidence of that when they look at these coronary, or look at the aortas of these mice that were treated, they could see significant increases in the level of a very important efferocytosis receptor MERTK, which helps macrophages recognize apoptotic cells. When they took bone marrow derived macrophages and they polarized them to the various M1, M2 like phenotypes, and then gave them PA, all of these classes of macrophages had an increased capacity to do efferocytosis. And in part, this was because the PEA increased the expression of MERTK, and this was in a GPR55 dependent manner because GPR55 knockout macrophages were not able to increase their expression. So this helps us understand in part at least the pharmacological effects of PEA and OEA and why this may prove to be able to prevent cardiometabolic disease. And certainly our studies with this long-term administration support that if we can somehow keep the levels of OEA and PEA high enough, we can protect against cardiometabolic disease. But it also leaves us with a number of important unanswered questions. One is actually how to administer these types of levels in humans. We can talk about that in the Q&A. But also human disease is certainly far more advanced than what you see in rodents. It's longer established, and so we really need clinical trials to know if administration of PEA and OEA will be beneficial. But it also raises the question, if OEA and PEA are so protective, then why isn't the endogenous biosynthesis of these compounds protective? And is this really just a pharmacological effect? Is it important endogenously? And to answer that question, of course, one of the ways to do that is to do genetic deletion of the various biosynthetic genes and ask, does this drive cardiometabolic disease? And we were particularly interested in this feeding-induced biosynthesis of NAPEs and NAEs, since it certainly mirrored what we were trying to do with the bacteria. But one of the challenges here is, again, like in humans, there's 6 different N-acyltransferases. So which one will we try to knock out in a mouse or such? And so we had a colleague, Wenbiao Chen, who does a lot of zebrafish work and certainly is looking at metabolic disease in zebrafish. And he suggested that we use them because it's much easier to do the knockouts in them. First, we had to ask the question, well, do zebrafish even have this feeding-induced biosynthesis of NAPE and NAEs like mammals? And so to test this, we fasted zebrafish for 48 hours and then split the group in 2 and kept half the group fasted, refed the other half, then collected their brains and intestines and measured the NAPE and NAE levels by mass spectrometry. And what we saw was there were very nice increases in intestinal NAPEs in response to refeeding, as well as intestinal NAEs. There was maybe a trend toward an increase in the brain NAPEs, but no change in the brain NAEs. So indeed, zebrafish, like mammals, have this feeding response. We then wanted to go through and knock out the various calcium-independent N-acyltransferases. Originally, so Wenbiao went and looked at the genome of zebrafish, and originally there were 3 plats were identified, but by using homology to human plats, he actually was able to identify 2 additional plat enzymes, which we called, because they clustered together with plat 1, we called plat 1L1 and plat 1L2. And when we looked at where these were expressed, they were expressed in the intestine, as well as other tissues. If we just did did the knockouts and just looked at NAPE levels in the whole body of the zebrafish larvae, we saw that both the 1L1 and 1L2 had significant decreases in NAPEs, and so we focused on those and looked at the effect of knockout on the feeding-induced NAPE response. Again, wild type have a very nice response to refeeding, but the plat 1L1 knockouts essentially have no response to refeeding. They don't make any NAPEs when they're refed, whereas the 1L2, there was a response. And so we, of course, then wanted to ask, well, what's the effect of knocking this out on metabolic disease? And so when we look at these knockout animals, they had increased body weight. And this is just being fed a standard flake diet. There wasn't a change in their body length, so bone and muscle seemed to be fine, but there was an increase in the body mass index indicative of an increase in fat accumulation. And there was also increases in their blood glucose, suggesting at least early hyperglycemia. So knocking out this feeding-induced NAPE response does at least induce moderate metabolic disease. But this leaves us with a lot of unanswered questions. Is plat 1 the N-acyltransferase responsible for the feeding-induced biosynthesis in mammals, or is it some other enzyme? And if we were to do these in atherosclerotic prone mice, would we see increases in the various diseases? So we don't know the answer. What about knocking out NAPE-PLD? So most of this work, it come from Patrice Caney's lab and Sergei Luque's lab. So they've done adipose-specific knockouts, and in that case, you see an increase in plasma cholesterol and triglycerides and ceramides. Interestingly, there's no increase on food intake, but there's an increase in adiposity and liver triglycerides and glucose intolerance when you put them on a high-fat diet. Now, if you knock out NAPE-PLD in the intestine, you do see the increased food intake, at least after initial exposure to a high-fat diet. You do have increased adiposity and hepatosteatosis, but there's no effect on glucose tolerance. If you knock it out in the hepatocyte, you see increased adiposity and liver triglycerides, and you see increased insulin resistance, again, on a high-fat diet. In the brain, you have interesting responses. So I think to summarize what you see there is that there is an enhanced reward response to highly palatable foods. So there's a strong preference there, but there's not a significance in difference in body weight or body fat. And this seems to be because even though there's an increase in food intake, there's actually an increase in spontaneous activity. That sort of counterbalances this. Now, my lab is very interested in what is the effect of knocking out NAPE-PLD in immune cells. And one of the reasons that we've been very curious about this is, again, going back to Sabine Stephan's lab's work, where they were doing the PEA injection in the ApoE mice. One of the things they observed was there were marked decreases in NAPE-PLD expression in the spleen and bone marrow of these mice as you fed them a high-fat diet. And this wasn't limited to humans. If you look in human coronary arteries, those with plaques have reduced NAPE-PLD expression compared to arteries without plaques. And those with unstable plaques, so those that have, essentially likely to have impaired efferocytosis and greater necrotic core, have the greatest reduction in NAPE-PLD gene expression. And they showed the coronary artery that NAPE-PLD was in macrophages and smooth muscle cells. So this led us to ask the question, what is the effect of deleting NAPE-PLD on efferocytosis and such? And I just want to say, one of the other reasons that we were also interested in deletion of NAPE-PLD is because we'd also been doing work with some other N-modified PEs. These are those that are generated when lipid peroxidation generates reactive lipid aldehydes. These can react with the nitrogen of PE to make these N-aldehyde modified PEs. And we've shown that these are pro-inflammatory. And we knew that if you had just had the modified ethanolamine, these are far less active. So that led us to hypothesize that if NAPE-PLD could actually act on these N-aldehyde modified PEs, it would serve to inactivate these. And so that would be an additional way of blocking inflammation in addition to their formation of these anti-inflammatory and pro-resolving NAEs. And I'll just go through this quickly, but so postdoc in my lab basically showed, generated the various N-aldehyde PEs, and some of these, like the 4-hydroxynonenal form of, is as good of a substrate for NAPE-PLD as the N-palmitoyl PE. So they are actually very good substrates. And so, again, we think that part of the effect of losing NAPE-PLD might be because you're not able to clear these pro-inflammatory N-aldehyde modified PEs. So what is the effect of NAPE-PLD deletion on efferocytosis? Well, you get significant reductions, and this is using 2 different strains of NAPE-PLD mice. We'd also developed small molecule inhibitors and activators of NAPE-PLD. This is just showing that we could cause these changes in the bone marrow-derived macrophages. So VU534 is one of our activators. Biothionyl is one of our inhibitors. So biothionyl phenocopies the deletion of NAPE-PLD. It reduces efferocytosis, whereas our activators increase the ability of macrophages to do efferocytosis, and an inactive compound does not have that effect of increasing. This is NAPE-PLD specific because if you look at knockout animals, again, if they're just treated with a vehicle, you see a reduction in their capacity to do efferocytosis. It's true with the activators. The wild type you see increase efferocytosis, but there's no change in the knockouts. What's driving this? There are changes in a number of genes, and if you do go analysis on these genes, it might not come up as a clear efferocytosis signature, but when you look at some of these genes, like ornithine decarboxylase 1, which catalyzes the first step in polyamine biosynthesis, recent data suggests that loss of ODC reduces expression of MERTK, it causes defects in efferocytosis in vivo, and makes it less able to do atherosclerosis regression. Similarly, lipin-1, which is a phosphatidic acid phosphatase, its deletion of myeloid cells increase in the necrotic core area in plaques. Hemoxygenase 1, its deletion makes macrophages secrete more cytokines, but also inhibits their ability to clear apoptotic red blood cells. So, we need to do more work on this, but this seems to just suggest why the loss of NAPE-PLD may be important. And also some preliminary data. We don't have NAPE-PLD specific, myeloid specific knockouts, but we can do essentially the equivalent by using the NAPE-PLD mice and doing bone marrow transplant into the LDL receptor mice. And what we see is if you do transplant from the knockout mice into LDL receptor mice, you have greater atherosclerosis compared to transfer of wild type macrophages into the LDL receptor mice. So indeed, deletion of these enzymes can at least cause moderate increase in cardiometabolic disease. So to reach the end here, I just kind of want to summarize what we do know and what we don't know about the role of these biosynthetic enzymes in protecting against cardiometabolic disease. So we do know that many stimuli do invoke their biosynthesis, but the specific enzymes that are involved in the precise mechanisms are very poorly characterized. We know that pharmacologic administration of PEA and OEA exerts substantial protective effects. If we could give these chronically to humans, they have the potential to be very protective, but we need to actually do those clinical trials. We do know that obesogenic and atherogenic conditions reduce NAPE-PLD expression, but we don't understand the mechanisms behind that. We do know that genetic deletion of some of these enzymes is sufficient to induce cardiometabolic diseases, but we really need much more extensive characterization. And small molecule activators of NAPE-PLD can enhance the efferocytic capacity of macrophages in vitro, but whether that would have any benefit in vivo needs to be determined. Finally, I just want to conclude by thanking the members of my lab who did these many experiments, as well as my many collaborators.
Thank you very much. Brilliant presentation. The first one, do you have any idea if we look, let's say, on the structure of NAPE, where they would reside in the cells?
So you're talking about NAPE-PLD specifically?
Yeah. I mean, with the 3 acyl chains, they would be quite hydrophobic, right?
Yeah, so I mean, the notion is they're definitely sitting on membranes, but if, in all of our studies and what other people have done, they're sitting on a lot of different membranes. And one of the things that we're interested in is, does that localization change at all with different stressors? Because we don't really know, but it seems like it ought to, but we don't know that. So a big unanswered question.
So you don't see localization with lipid droplets for them?
No, and I've asked people that have tried to do proteomics on lipid droplets, and because I'm always thinking that NAPE-PLD should be there, but at least so far, none of them say there's any evidence for it. But yeah, so.
Okay, there is a question from Jason.
Hey, fantastic talk. Thank you so much. So some of the data you presented reminds me of ferroptosis in that there's some intersection with lipid peroxidation byproducts, and also that these enzymes or these pathways are involved in ischemia. So I was wondering if you think that part of the role of NAE is in resolving this sort of proferroptotic stress.
So the answer to that is yes. My postdoc Reza has actually been trying to do experiments with RSL3 and such. We don't have data that we can share on that at the moment, but I definitely believe that there is likely to be a role.
Awesome, thank you. Good luck with those experiments.
No, that would be, of course, very interesting to see. Is there any staining to track where they accumulate in cells and any certain track of downstream metabolites?
Yeah, so that's kind of a challenging question. So one of the things, we do have a fluorogenic NAPE that we add to cells that we can measure NAPE-PLD activity, and so we can see where that is being formed and kind of follow it. So that gives us some hint of where NAPE could be converted to NAEs, but it doesn't really show us like where the NAPE itself would actually be generated. Theoretically, you could use isotope tracing and then do separation of the organelles. Of course, that would have to assume that there wasn't equilibration between the organelles as you're doing the separation. So that's, I think, one of the the big challenges is trying to be able to really understand at that subcellular level what is happening, because I really do think there's a lot of interesting things that are probably happening there.
Absolutely. So many things for me. And just to get an idea, I mean, you showed a few slides where there were quantities, right? But those, let's say, species are very low, right?
Yeah, I mean, I think like if you think about NAPEs in general, you're talking about 0.1% of phospholipids, right? I mean, these are bioactive lipids. They're not bulk lipids. I mean, so location does matter. Being by the receptor would have to matter. They're not. I mean, I think some people put forward the idea that the NAPEs help stabilize the membrane, and that may be true, but they're very, very small percentage of the phospholipids in a membrane. So I'm not sure that is really that likely.
In terms of the distribution, let's say, of the enzymes or of the synthesis, right? So that would happen mostly on the ER membrane. I think you had a slide on that.
Well, again, I think that's an interesting question. Again, NAPE is widely distributed. I mean, the acyltransferases probably. Again, it's amazing how little we actually know about where they are. I mean, I think it's reasonable to think that they're definitely made in the ER, but they may also be made in the mitochondria and maybe at other sites as well.
And you also mentioned on the effect, let's say, on the lesion necrosis, yeah, reduction of the effect over there. And then you also mentioned that you have an increase in the collagen content, right? So
Yeah.
does it mean it's a bit like profibrotic in this context? Because in the same animals, I guess in the liver, you had a reduction of fibrosis, right?
Yeah, I mean, so I think it's an interesting question. Of course, I mean, we know overall that, I mean, one of the responses as you develop atherosclerosis, if you is to have that fibrotic cap formed. I guess maybe another way of thinking about it is not so much maybe that the PEA is driving the formation, but it's preventing the thinning that comes from when you have the necrotic cap. But it may be. I mean, certainly our evidence isn't so much that it would drive that fibrosis per se. But I think it's an open question.
So we have a question from Marius.
Sean, great talk. I'm fascinated by the bacteria producing NAPEs. Can these mechanisms or these molecules be used to escape resistance cells surveillance, so that the signal, okay, we are calm here, don't attack us, especially, well, if E. coli produces, it might be used to modify the host response. But then there might be also other bacteria living on the skin that might also utilize this to keep the host's resistance in balance. What do you think about this idea?
So I mean, I think this is one of the challenges. I'll be straightforward here that we've kind of moved away from the bacterial delivery mechanism because we really don't know how we can move it to humans, and that is one of the concerns. Is if you had horizontal transfer into pathogenic bacteria and it was essentially turn off. I mean, it would have to, of course, into the host, but that can certainly happen. And then if it had captured that system, would it be able to protect itself? And so, I mean, I think that is one of the concerns with using the bacterial system in humans. I mean, we didn't have evidence for that in mice, but I mean, a challenge there is none of those experiments are done for years and years. And so, you know, obviously in humans, the opportunity for horizontal transfer would persist for a lot longer. So you'd absolutely have to have passive containment methods to ensure that you didn't have horizontal transfer. And that's frankly one of the problems that we didn't feel like we could solve.
My question is based on observation I've recently seen in my data. There is something that is similar to NAPE in extracts from Cutibacteria, so clinical isolates of bacteria that reside on the skin and are responsible for acne. And in our discussion, one of these hypotheses would be, well, they can produce this to just to escape the surveillance of the host of our immune system. So there might be already bacteria potentially pathogenic living in us or on us, which can synthesize those NPEs. And
well,
maybe
yeah, I mean, and I should say, even before we started, I mean, we knew E. coli could make small amounts of the N-acyl-PEs, and others can as well. But those are relatively small compared to what the engineered ones were producing. But certainly, you know, in your case, I mean, if they were making large amounts, I mean, you might expect that they would, you know, that is a response of the bacteria to protect themselves.
Okay, thank you very much. It was a very interesting talk.
And we have one more question from Han. My very last question. We're running a bit out of time. The question is, did you or any other groups see any effect on the adipocyte browning?
So I know that we tried to look at that and we didn't see an effect, but in our bacteria things, but there definitely have been people who were looking at the effects. So like in some of the Petrus Caney's work, that they were, and what I'm recalling is they did actually see a browning effect and expression like of UCP1. I'm trying to recall the exact details, but I do think that was something that they saw when they were doing the adipocyte deletion and such. Okay, so Hans is asking about the monoacylglycerols and proteins affected by NAs. So one of the things that, so Heather Bradshaw has done some really nice work with lipidomics and showing that like in the NAPE-PLD knockout, you have changes in many different types of lipids. And one of the things that is known is that some of these NAEs actually will work on DAG lipase and increase its activity. And so you can have direct downstream effects on the 2-AGs from producing the NAEs. Now, whether there's like protein interactions, you know, there isn't evidence for that, but definitely there is evidence that the NAEs themselves may be acting on these other pathways.
Thank you very much, Sean. I think we have to round up for today. So thank you very much again for this very amazing talk. Thank you all very much. Have a good day, evening, and see you soon. Thank you, Sean, again.